Before the acceptance of germ theory, the predominant belief was that diseases were caused by miasma or bad air, imbalances in the body's humors, or spontaneous generation. This lack of understanding of disease causation limited the effectiveness of medical treatments and public health measures. In the late 17th century, Dutch scientist Antonie van Leeuwenhoek used his handcrafted microscopes to observe microscopic organisms, which he referred to as animalcules. While he did not connect these organisms to disease, his observations laid the groundwork for future discoveries in microbiology. In the 17th and 18th centuries, Italian scientists Francesco Redi and Lazzaro Spallanzani conducted experiments that challenged the theory of spontaneous generation, showing that life did not arise from non-living matter. Their work suggested that living organisms came from other living organisms, hinting at the possibility that microbes could cause disease. French chemist and microbiologist Louis Pasteur is often credited with providing definitive proof for the germ theory of disease. In the mid-19th century, Pasteur conducted experiments that demonstrated that microorganisms were responsible for fermentation and spoilage in food and beverages. His famous swan neck flask experiments showed that sterilized broth remained free of microbial growth unless exposed to air containing microbes, disproving spontaneous generation. Pasteur extended his research to study infectious diseases. He developed vaccines for diseases such as anthrax, chicken cholera, and rabies by demonstrating that weakened or killed pathogens could induce immunity. These breakthroughs not only provided effective means of preventing these diseases, but also supported the idea that specific microorganisms were responsible for specific illnesses. German physician Robert Koch further validated germ theory by identifying the specific bacteria that caused anthrax, tuberculosis, and cholera. Koch formulated a series of criteria, known as Koch's postulates, to establish a causal relationship between a microorganism and a disease. The microorganism must be found in all cases of the disease. It must be isolated from the host and grown in pure culture. It must cause the disease when introduced into a healthy host. It must be re-isolated from the experimentally infected host and identified as the same organism. Koch's discoveries and methods provided a systematic approach for identifying pathogens and understanding their role in diseases. This led to significant advancements in diagnosing, treating, and preventing infectious diseases. The acceptance of germ theory revolutionized public health practices, emphasizing the importance of hygiene and sanitation. Measures such as improved sewage systems, water treatment, and sterilization of medical instruments were implemented to reduce the spread of infectious diseases. Inspired by germ theory, British surgeon Joseph Lister introduced antiseptic techniques in surgery, using carbolic acid to disinfect wounds and surgical instruments, dramatically reducing post-operative infections and mortality rates. The development of vaccines gained momentum, with scientists building on Pasteur and Koch's work to create vaccines for a wide range of diseases, further reducing the incidence and impact of infectious diseases. Germ theory spurred the growth of microbiology as a scientific discipline. Researchers began identifying pathogens responsible for various diseases, leading to the development of targeted treatments and prevention strategies. Medical education underwent a significant transformation, incorporating microbiology and germ theory into the curriculum. This shift led to more scientific and evidence-based approaches in diagnosing and treating diseases. The germ theory of disease, established through the pioneering work of scientists like Louis Pasteur and Robert Koch, revolutionized the understanding of disease causation. By demonstrating that microorganisms were responsible for many infectious diseases, germ theory provided the foundation for modern medical and public health practices. The resulting improvements in hygiene, sanitation, antiseptics, and vaccination significantly reduced the burden of infectious diseases and transformed medical science, paving the way for advancements that continue to save lives and improve health worldwide.